Hello everyone, I'm Dr. Nikolas Pontikos and I'm a PI supported by Moorfields Eye Charity. I'm based at UCL Institute of Ophthalmology and Moorfields Eye Hospital, and today I'm going to talk to you about my research project Eye2Gene. So I'm a computer scientist by training with an MSc in bioinformatics and a PhD in statistical genetics. My team's research is in the area of genetics, medical imaging, and artificial intelligence. And I've been working with ophthalmic genetic specialists over the last 5 years. So as you've probably heard, inherited retinal diseases, or IRD for short, are caused by mutations in over 250 genes. These genes make proteins that are expressed in different cell types, and because of this difference in cell-specific expression, IRDs tend to have characteristic patterns of retinal degeneration, as you can see here, and these are detectable through imaging modalities such as autofluorescence. And hallmarks of IRDs include features such as area of hypo or hyper autofluorescence due to ellipsoid zone loss or accumulation of lipofuscin. So identifying disease-associated genes is really important for gene therapies such as Luxturna for RP65 retinopathy, as you are aware of. At Moorfields Eye Hospital, IRDs have been studied now for over 30 years. And in the last 20 years, we've been able to identify the IRD causing gene in over 4,000 patients and 135 genes. Here below, you can see the distribution of Moorfields IRD patients across IRD causing genes in adults and in pediatric cohort under 18-year-olds. It is immediately obvious when you look at this that both adults and children have in common the most prevalent cause of IRD, which is ABCA4, which is associated with Stargardt disease, as you've heard from Dr. Porto. However, we see that for the rest of the genes, the distributions are different. And in particular, for the second most prevalent gene in adults, it's USH2A, which is responsible for retinitis pigmentosa and hearing loss. And in pediatric cohort, it is the RS1 gene, which is associated with X-linked retinoschisis. So there's difference in gene distribution between adults and the pediatric cohort, really is for two reasons. One is because the age of onset of these genes differs. So for instance, USH2A associated disease is usually slow progressing and only noticeable in later life. And the other reason is because Moorfields is primarily a hospital for adults with eye-specific conditions. And so syndromic pediatric patients are often seen in other hospitals, specialist pediatric hospitals such as Great Ormond Street. However, one important thing is that these patients only represent 70% of all the IRD patients seen at Moorfields. So there's over 1,000 patients for which we do not have a genetic diagnosis, even at Moorfields, which is one of the leading centers for IRD studies. So this is an overview of the genetic diagnosis process in the UK. So how do patients actually get referred to Moorfields Eye Hospital? So based on a prevalence of 1 in 3,000 IRD in the UK has a population of 65 million. So we estimate there must be around 25,000 individuals which have an IRD in the general population. Some of these individuals will be seen by an optometrist, and in some cases, these optometrists will be able to refer patients to eye hospitals if they detect a disease. Patients will be examined there by a multidisciplinary team, which includes clinical specialists, genetic specialists, and bioinformatics specialists. And they will perform genetic testing based on the suspected disease. And the types of genetic tests can be either a single gene or gene panel, and these are available on the National Health Service of the UK, or now more and more whole genome sequencing, which is used to be just for research, but now is going to eventually be rolled out as part of the National Health Service as well. This whole process can take more than 5 years, and in the end, it can be very difficult to pinpoint the actual disease-causing variants because there can be hundreds of possible disease-causing variants per person. In most eye hospitals in the UK, we estimate that generally the diagnostic yield is 60%. So based on that number, we estimate that there must be around 10,000 patients at least in the UK that do not have an IRD, but do not have a genetic diagnosis. So the two reasons really for this limitation in the diagnostic rate is firstly detection. So there's few optometrists which are capable of identifying and hence referring IRDs. And also at the hospitals, there's also a shortage of expert teams capable of identifying disease-causing mutations. So these bottlenecks, they can be addressed with AI, and that is the goal of my Eye2Gene project. So the short-term goal really is to start in the hospitals with the data available in the hospitals and to see whether we can improve the diagnosis yield and process there. And this is through guiding genetic variant interpretation, which will in turn mean ordering the right genetic test and then increasing the diagnostic yield at a decreased economical cost. And the sort of long-term goal is to improve the screening of IRDs in the community and maybe at some point move genetic testing to the community. So for the for the short-term goal, which is what I'm going to be focused on today, we want to offer an AI algorithm that I have called Eye2Gene that will offer a complementary diagnosis that can be used by the multidisciplinary team in order to assist their genetic diagnosis. The way we're going to achieve this is to use an AI tool known as a neural network, and more specifically a convolutional neural network, which will be able to identify the most likely disease-causing gene based on a retinal scan. So in order to develop and train such an algorithm, we need large amounts of genetics data with imaging data. Thankfully, at Moorfields, we have one of the largest data sets of IRD patients, along with imaging. And these are the numbers we have. And however, one thing, though, is that even in this large data set, because of the general the skew in the gene distribution, beyond the top 50 genes, most common genes, we have less than 5 individuals per gene diagnosis. And this is too little to train a neural network. So we will focus here on the top 50 genes. And here you can see these are the top 20 genes and the sort of numbers we have. So in order to train this neural network, this convolutional neural network, we split the data set into training and test data sets. And we keep one data set, we withhold one data set for benchmarking. And we use a specific convolutional neural network architecture known as a inception network. Following the training, here are the results that we obtain. And what you're seeing here is what's known as receiver operating curves. And this is how we assess the performance, prediction performance. And the metric that we are interested in maximizing is the area under the curve, under the blue curve. So for instance here, ABCA4 has a high performance, whereas this gene has a much, CDH23 has a much lower performance. So this area under the curve, or AUC for short, is proportional to the number, as you might expect, to the number of training examples per genes. So genes which have a large number of training examples like ABCA4 have a better performance than ones which have a lower number of training examples. But it's also depends on the phenotypic heterogeneity of some genes. So BEST1, for instance, which causes retinopathies, they are quite phenotypically heterogeneous, and hence the performance is not great, even though they are quite common in our data set. From this neural network, we can run something called an analysis tool called an attention map, and this allows you to highlight the part of the image which were used by the neural network to reach a correct classification. So as you can see here, it seems to highlight regions of atrophy, so it seems to be working well. At least it is highlighting relevant features. So the performance is good, is not bad, and these results are promising. We've only been able to do the top 50 genes, but for the lower tail of those genes, well, we're not getting very great accuracy. For instance, in the rarer genes, for AHI1, the performance was not very good, and those with high phenotypic heterogeneity, such as BEST1. And also, probably this neural network will not generalize across different medical devices. So if, for instance, it's been trained on a Heidelberg device, but it probably wouldn't generalize well on, be applicable to TopCon imaging. So basically, what's the solution to these problems? Well, as you might think, it's just, well, more data, really. And more data is great, but the challenge is we are working with rare eye diseases. So we have to really try and find this data. And one way of going about it is to set up these sort of material transfer agreements between hospitals to get data directly from hospitals. And that's a good way of doing things. However, these processes can take months and even years, and it's difficult to arrange these. You know, within a country, it's okay, but even like between countries, there's all different rules and laws, and this can be quite complicated. Another approach, which we've been exploring, is going to patients and asking for the patients to provide their data. And this works quite well for rare disease patients because they're quite motivated by being kept up to date with research in the case in the eventuality where a treatment would be developed for their disease. So they really want to share the data to stimulate research and development of treatments. And so this is the MyEyesight project. And MyEyesight is basically an app that we've developed in collaboration with a commercial partner known as Loft Digital. And Loft Digital and us, we've been funded by Health Data Research UK and the UK Research, UKRI, Research Innovate UK. And what MyEyesight is really is it's an app on your phone, and it allows patients to request their data from the hospital and link it from different hospitals, because if you're seen in different hospitals in the UK, your data is not automatically joined. You have to go and manually request it from different hospitals and centralize it to be able to join it. And it allows also patients to contribute more to their own data. So for instance, they can report patient-reported outcomes or quality of life, answer questionnaires. And they can also link their data to other members of their family, which is quite important for genetic inherited conditions. So really, it allows them to have data as an asset for long-term personal use. And in the context of Eye2Gene, it's really important because it allows them to share their imaging data directly with Eye2Gene and other research projects. They can also obviously share their data with doctors in other hospitals and with whoever they decide. So really, it's the bottom line is it's about empowering patients, and it's really, from our work and our survey, it's something that's really well received by patients and that they want. So this is what's being developed. You can go and have a look at our website to keep up to date and sign up to the newsletter to keep up to date with the latest developments. There is an app. It's not released on the App Store there yet, but it will be at some point, I imagine, over the next year. So to summarize, AI approaches such as Eye2Gene do have the potential to democratize expert knowledge for rare eye diseases such as inherited retinal diseases. However, for them to be developed, they need to have a good and global view of data, health and disease. And this is why really sharing, especially for rare eye diseases, is really essential. And this is a challenge, and we can try and do it between hospitals, but with digital technologies, now patients can help us in this effort, and they can help us by retrieving their data from hospitals and sharing it on their own terms to stimulate research. So thank you very much. I hope you found this talk interesting. Obrigado. And I'll leave you with some references if you're interested to go and have a read on the Eye2Gene website or the MyEyesight page. Also some references to to the IRD gene list that you probably are aware of. IRD counts, which is an interesting project looking at the prevalence and the health economic impact of IRDs, and relevant publications. Thank you very much. I'll take any questions now.